is the slide and not the presentation tools?
Yes.
Okay, thank you. Okay, 1 second. So my name is Tara Dolan, and like Gary said, I work for the Massachusetts Division of Marine Fisheries. I'm here to talk to you today about the stock structure of winter flounder. This is a continuation of work conducted at Stony Brook University in Long Island, New York, as part of my dissertation. But when I was lucky enough to get a job at Mass DMF, they allowed me to continue my research on winter flounder, which you will see is a fascinating study species with complex stock structure and diverse behavior. First, a quick primer on winter flounder. They are a right-sided flatfish that ranges from Virginia to Maine in the United States and north into maritime Canada. In the United States, it's managed as 3 different stocks: the Gulf of Maine, Georges Bank, and Southern New England Mid-Atlantic, which throughout this talk I'll be abbreviating as GOM for Gulf of Maine, GB for Georges Bank, and SNE for Southern New England. Winter flounder are an iconic species for Massachusetts, supporting a long history of commercial and recreational fishing. Quincy, Massachusetts famously proclaims itself the flounder capital of the world. So you can see that this species is super important for this area. All 3 stocks have declined in abundance since the 1980s, and even though exploitation rate has remained low, we really aren't seeing a rebound in biomass, especially for the Southern New England stock. In Southern New England, which is the stock we'll mostly be talking about today, the stock has not been subject to overfishing since 2010, and yet it has remained in an overfished state for over 10 years. The only reason it came out of that state in the most recent stock assessment in 2022 is because we sort of changed the frame of reference for what productivity we believe is even possible for this stock. So it's still in a pretty depleted state, regardless of status. Now that we've identified the problem, let's back up a bit to the concept of a stock. One reason we divide fish up into stocks is to audit fish populations to understand the effects of fishing. Stocks, by definition, are discrete units with homogeneous internal dynamics. Necessarily, they can be exploited independently. Maybe they comprise different fisheries, or they could belong to a mixed fishery, but in that case, catch could be reliably assigned back to its stock of origin. Stock delineation has management implications because population components could have different productivity. They could experience different fishing or natural mortality. They could comprise different fisheries. It's critical that we get the stock units right because mischaracterizing trends in stock structure can lead to mischaracterizing trends in productivity, which could have a ripple effect through management. So why are some fisheries slow to recover from collapse, other than that they're continuing to be overfished? 3 reasons, among many, that we're going to grapple with here are: 1, the fishery has changed; 2, the environment has changed; and 3, most importantly for today, the stock has changed. And of the many ways the stock composition can change, there are 3 we're going to talk about today are loss of genetic diversity, loss of life history diversity, and loss of connectivity. Diversity loss is especially bad because, much like an investment portfolio, life history diversity leans to stability over time in a variable environment. And this is only provided there is sufficient connectivity among populations. The portfolio effect, there's actually supposed to be a blue and a gray line there. I don't know what happened. But the point is that even if individual populations are showing different trends, if there's connectivity between these populations, these depleted population components can rescue the other ones if there's enough connectivity, and the long-term average will remain stable. From a genetic standpoint, connectivity is gene flow through reproduction. On the one hand, barriers to gene flow can prevent reproductive mixing and can cause genetic divergence in population structure. But there's also barriers to gene flow. Barriers to gene flow could be structural, like a dam, or they could be behavioral. Here, there it goes. They could be structural, like a dam, or they could be behavioral. Like, for example, groups might not be reproductively mixing and exchanging genes because they might use the same area, but they might be spawning at different times, like this group that spawns in the spring and this one that spawns in the fall. Or they might not be barriers at all, but simply forces which dampen gene flow, like ocean currents, which might be semi-permeable, but reduce gene flow, or physical distance. A pattern when gene flow decreases with distance is called isolation by distance. So we're going to talk now about patterns of connectivity in winter flounder. Winter flounder are so named because they return to coastal bays and estuaries to spawn in the spring, to spawn in the winter and early spring. Young of the year hatch in bays during the spring, and after a short pelagic duration, they settle on the bottom, and they use selective tidal stream transport to remain in the estuaries. So here you can see the adult migrating in, mating, creating these eggs, and the eggs are staying on the bottom. The larvae are staying in the system, and they're staying in there for 2 years, approximately, maybe more like 18 months. So let's remember that gene flow requires reproductive mixing. If we are to assume that these fish have true natal philopatry, if they're truly returning to the bay that they were born in, we would expect to see strong population structure between bays, because we're assuming that the eggs and larvae are retained in the estuary. So this would be both a physical and behavioral resistance to gene flow. So it's physical because the eggs and larvae are in the bays and they're returning back to the bays, and it's behavioral because they're returning back to those bays that they were born in. But the evidence for differences in genetic structure between proximal estuaries is mixed. These two studies on the left were both of compared estuaries that were close to each other. Crivello found larval advection into estuaries, mixing things up and not really seeing genetic differentiation between proximal estuaries. But Buckley found an incredible amount of genetic structure in a very small area. And then Wergen tried to repeat this, but only found differences when pooling samples across stock area. So found genetic differences at the stock level, but not in between estuaries. Then some studies on Long Island, including O'Leary et al., found some differences between bays, but only weak structure. And then the study that we did found similarly some structure and differences between bays. So why are these studies differing? These differences could be attributed to the spatial and temporal scale at which samples were pooled. They could be attributed to the choice of genetic marker that was used. And they could also be attributed to differences in statistical power. And this only highlights the need to understand the power and the limitations of different genomic tools. So digging deeper into that last study on Long Island bays. So first, to orient you, Wright's FST is one of the ways we measure genetic structure. It's a measure of differences in allele frequencies between populations. So here we have red and blue flounder, which indicate Long Island bays with significantly different FST. So we're not seeing that one of these bays is a totally different color. We would expect to see under strict natal philopatry, and we're not seeing a pattern of isolation by distance, which is where groups that are geographically close to each other are more closely related. And that's the pattern that we might expect to see if structure were controlled by larval dispersal. Instead, what we're seeing is likely a pattern of some natal homing with straying by adults. So it's an adult movement factor of adult behavior that might be creating these patterns. And indeed, this is supported by some other studies on the stock and their behavior. It's very important to realize, this is my soapbox, that genetic connectivity is not the same thing as demographic connectivity. Whereas demographic connectivity is immigration on the scale that influences population dynamics. So ecological connectivity, demographic connectivity, that's on the scale that's influencing population dynamics. It does not take many individuals to maintain genetic connectivity. So studies where you take genetic samples versus studies where you mark recapture individuals are answering slightly different questions, but they're both really important to understanding stock structure overall. And since it's going to come up again, just a reminder that Wright's FST is one measure of genetic connectivity. It's a measure of differences in allele frequencies between populations that can indicate genetic structure. So we have FST on the y-axis and the number of immigrants per generation on the x-axis. And you can see that with less than 10 immigrants per generation, you're spreading advantageous alleles. You're more immigrants per generation, you're avoiding the effects of inbreeding. So what several studies of winter flounder show on Long Island and across the stock area show is that we're seeing FST at very, very low levels, and we're seeing enough immigration to maintain similar allele frequencies and reduce genetic structure between bays. But to see how this low level of population connectivity can have meaningful effects for diversity, we need only compare our study to a previous study that covered many of the same locations, which was O'Leary et al. 2013. And though we can't directly compare the studies, we did find that in just 6 years after O'Leary's study, we saw lower levels of inbreeding, including no excess homozygosity, lower internal relatedness, and a higher number of effective breeders in all locations. And this was contemporaneous to a slight uptick in spawning stock biomass in southern New England. And this is, but correlation causation, who knows? But this is good news because inbreeding is linked to lower survival, lower reproductive success, and lower disease resistance. So on this sort of like number line axis, y-axis here, you can see the internal relatedness at the O'Leary et al. Study, which was doing its sampling in 2010, showed internal relatedness where individuals from the same bay had about the internal relatedness that you would expect for half siblings. So a lot of inbreeding going on. And then our study, which is the little green flounder, was quite reduced from that. So we've already pulled apart one aspect of this traditional paradigm of winter flounder. Now we're going to take on some of the others. Recently, we found evidence that winter flounder do a lot of things that they're not supposed to do, under the assumption of natal patry, that is. For example, they spawn offshore, not just inshore in the estuaries, and this is on Long Island too, not just in the Gulf of Maine. They show partial migration, which is a behavior in which part of the population is undertaking seasonal migrations between the bays and offshore, and the other part of the population is remaining resident inshore. And an acoustic telemetry study, different acoustic telemetries, I'm actually referencing two different ones here, show that between 20 to 45% remain resident, depending on the location. And then multiple tagging studies from Long Island, New York, and New Jersey show that about 10% of winter flounder are visiting bays other than their natal estuary. And this is comporting with our observations of genetic connectivity. So studies have documented partial migration and offshore spawning. And we know that partial migration is one form of intraspecific diversity that has profound ecological consequences. For one, as we said earlier regarding the portfolio effect, having diverse population components can lead to stability, and this is the portfolio effect. But on the flip side, these diverse population components are exposed to different pressures. They might have different fishing pressure, be exposed to different habitat degradation. The key is whether it's possible to erode this diversity by disproportionately removing one component. So it's really important to have some level of connectivity between these components so that they retain their individuality, but also can rescue each other if one is depleted. So here we're showing several different hypotheses for how partial migration could be maintained in winter flounder populations. It could be the result of separate lineages where individuals are born either migrant or resident, and the migrants are doing their migration and the residents are staying in the bay their whole life. The second option, option B, is that resident or migrant status is determined in early life history. Maybe it's epigenetically controlled, but not genetically controlled. Maybe it's socially entrained. Maybe it has to do with different environmental conditions during early life history. These are all possibilities that have been documented in other species, but we're not really sure for winter flounder yet. So they might decide, they're not deciding, but it might be determined in early life history whether they're migrants or residents. And then finally, another option is that migration status could be flexible throughout life. It might be a strategy that they employ depending on conditions, depending on social entrainment, so depending on which groups they encounter, or it might be an ontogenetic transition or differences in sex or something like that. So maybe with ontogenetic transition, maybe they're migrating as large adults, but not as small individuals, or maybe males are not migrating. These are all things that we have to learn more about and can learn more about. So one thing that was interesting is that a previous study by Siski et al. In Canadian Journal used otolith microchemistry to categorize fish across 3 stock areas into either bay versus ocean nursery or resident versus migrant adults. So they did bay versus ocean nursery by looking at the magnesium calcium ratio in the fish's otolith in their early life history. So they drilled down to the core of the otolith and they just looked at the magnesium to calcium ratio, whereas enrichment in magnesium might be a proxy for exposure to hypoxia in early life history. And then when they wanted to look at resident versus migrant, they looked at the whole otolith, so looking at the adults, the adult life history, and enrichment in strontium is a proxy for more oceanic conditions. So I had genetic information for some of the fish that Siski sampled because he was my lab mate, we worked together. So this seemed like a good opportunity to ask whether there was a genetic association with spawning and migration behavior, and whether there was a nursery, whether the nursery was the ocean or bay. So here we have the different categories are bay migrant, bay resident, so that's born in a bay, later a migrant, ocean migrant, and ocean resident. So the genetic studies that I mentioned, we're going to talk about methodology now. The genetic studies that I mentioned earlier were for the most part done with microsatellite markers. For this study, I used a different method to genotype individuals. This method is called restriction site associated DNA sequencing, or RAD-seq. And RAD-seq essentially randomly samples the genome. Each individual is randomly sampled at the same 1,000 or so locations across the genome. And we're comparing across individuals. And what we're comparing is slight variations in the genome called single nucleotide polymorphisms, or SNPs. SNPs are found throughout the genome. They're found both in the neutral part of the genome, which can tell us about neutral processes such as genetic drift. And they're also found, unlike microsatellites, which were used in the study that I previously mentioned, They're found in the part of the genome which is subject to selection. So we can learn a little bit eventually about selective pressures. So I get my SNP library from all of my individuals, and then I want to analyze it. And one method I use to analyze it is called discriminant analysis of principal components. So how this works is you're collapsing genetic variation across the different SNP loci into a PCA, into a principal components analysis. And then we're using a clustering algorithm. I used k-means clustering. And then from these different, with the k-means clustering, you can specify the number of clusters you're trying to cluster into. And then from this, each individual is assigned to a group with a membership probability. And this is what that looks like. On the y-axis, you have membership probability. On the x-axis, you have individual. And then you can see on the top x-axis that there's different stocks. And so the bars are individuals within each stock, and the colors are what cluster they belong to. So the first individual in stock A fully belongs to cluster 2, but the second individual in stock A, it's not, we don't really know. We're not entirely sure which cluster it belongs to, because the membership probability is not 100%. So the goal here is to use the number of clusters to tune into the signal that's coming from this data. So, for example, if migration or nursery status was caused by a single loci of large effect, like how migration timing is controlled by one gene in Chinook salmon, it would look like this. So here we have different stocks. We are giving them 2 clusters, and the dominant signal is just reflecting the migration propensity. So if we have Southern New England, for example, the stock A is 2/3 resident. You're seeing 2/3 of the colors are blue, and so that's showing that the signal that's coming out of this is the migration signal. Now, another way that migration behavior might manifest is through many loci of small effect throughout the genome adding up. For example, this is the case of a lot of environmentally associated changes like female interbrood period in some guppy species. So here, if we give it 6 clusters, you'll see a hierarchical situation where the migration signal might be present. So you're seeing different colors within stocks that have both migrant and residents. But you're also seeing this dominant signal, which is the different stocks are different colors. So you're seeing this dominant signal of stock differences, and then we're seeing the signal of migration versus resident. So if there were no discernible migration effect, you might see a pattern of isolation by distance, and here populations that are geographically closer will look more similar, but you won't see the migration signal at all. So here we're going to go on to the real data. So in the otolith study, we had 4 groups: ocean migrant, ocean resident, bay migrant, and bay resident. So I wanted to see if these different groups could be seen. The signal of these different groups could be seen. So what we're seeing is that when we give it 4 clusters, we're seeing this suggestion of hierarchical clustering within a stock. So we're seeing, for example, 2 different colors within each stock. Could that be migrants or residents, or could that be ocean nursery versus bay nursery? But the main signal here is that we're seeing strong differences between the Gulf of Maine and the 2 other stocks. So if we're migrants versus residents or ocean versus bay, what we are seeing here is we were looking to see if it was hierarchical, which could indicate many loci of small effect. We're trying to see if these differences within a stock are lining up to these designations of a bay migrant versus bay resident, for example. And what we're seeing is that that is not the case. We do not see that there's any clustering by migrants versus residents or by ocean versus bay nursery, but we are still seeing that stock differences. So then I wanted to give it 10 clusters because there's a total of 10 different categories, and see if we could draw out some of these clusters differently. And what we're seeing here is that the clustering algorithm clusters have absolutely no relation to the groups. Other than that, there are these still continuing to have these stock differences. So we're not seeing any signal from the interaction between migratory, contingent, and nursery, but we are seeing this like weird overlap between Georges Bank, quote unquote, Bay residents and Southern New England ocean migrants here, which is very strange. So Siski et al. Addressed this in his study. Yeah, here it is. He attributed the existence of Bay residents from Georges Bank, which seems difficult to believe because Georges Bank is out in the middle of the offshore waters here. How could there be Bay residents? But he attributed the existence of Bay residents to straying from Southern New England, and we did see that Georges Bank is very genetically similar to Southern New England, so I'm wondering if there's some sampling bias here. But they said in their paper that benthic hypoxia on Georges Bank could enrich the magnesium to calcium ratio, which could cause potentially a false Bay classification. So dig into this, that aside, to dig into the signal of the data a little further, I did a redundancy analysis, which models how genomic variation here is explained by geography versus the otolith microchemical signature. So this is basically an ordination. We're modeling the linear relationships among environmental, but in this case, we're using otolith microchemical instead of environmental data, and spatial predictors versus genomic variation. So first, we're going to look at a geographic model only. So this is only looking at geography. And what we're seeing here is that genomic differences are significantly explained by geographic distances. We see these blue, excuse me. Need a drink of water. We see these blue dots here from Southern New England are clustering differently than these green and red dots from Georges Bank in the Gulf of Maine. And these two little bars here are showing that the most significant divides are between the South Shore of Long Island and the Gulf of Maine. So that's sort of like the northern and some southern limits of our study. Like these three dots all the way up at the top are from northern Maine. So what we're seeing here is this like isolation by distance where individuals that were captured further away from each other are more different. And this is allowing us, this layer of the model is allowing us to account for spatial autocorrelation. So then I did a redundancy analysis, which is looking at the otolith microchemistry model. So we're looking at whether otolith microchemistry can explain differences in genetic variation. So we have our spawning migration class. Again, that's the Bay migrants, Bay resident, ocean spawned individual that was migrant, ocean spawned individual that was resident. And I used the strontium calcium ratio and the magnesium calcium ratio as explainers for this variation. And we did a model selection process. And eventually what came out, we also did, not shown here, but we did a partial RDA, which is controlling for that spatial signal, which was not significant. And what we're seeing here is that the otolith microchemistry, it's quite obvious, all these dots are on top of each other, is not, that does not explain any genetic variation. So their status is Bay migrant, Bay resident, ocean migrant, ocean resident is not is not showing up in their genomic data that we have found so far with our random sampling of the genome. So like a small portion of the genome. That doesn't mean that it might not be in other parts of the genome that we haven't sampled. And that's part of another project that we're going to be looking into in the Gulf of Maine moving forward in collaboration with UMass Amherst, which is very exciting. One of the great things about RAD-seq as a technique is that we can actually look at our focus, narrow our focus to look at the part of the genome which is under selection. And we do this with FST outlier analysis. So from the loci that are identified as being under selection, we see some indication of differences between stocks over here on the right. Oh, sorry, the left of the screen. You're going to see these green individuals from the Gulf of Maine that are sort of separating out from the blue and red individuals from Georges Bank in southern New England. So this is, if you recall, different than we found in the RDA. So we see Gulf of Maine, Georges Bank in southern New England here clustering together, whereas if you look at the geographic distances, we saw Gulf of Maine, we saw southern New England out by itself. So anyway, here we're seeing stock level differences here on the left. And then on the right side of the screen, we're looking at differences in the region of the genome that's subject to selection. So could these be differences created by different selective pressures that could be arising from different conditions encountered in early life history? If you're if you're born in an ocean, are you subjected to different selective pressures compared to if you're born in a bay? If you're an ocean migrant, if you're a migrant, are you subject to different selective pressures versus if you're a resident? Yeah, so that should be shown out in this clustering if that were true. But what we're seeing here is that we are not really seeing any clustering in the region of the genome that's subject to selection. So we did not find any indication of pattern in that case. So we've covered that young of the year produced by migrants or residents are likely from the same group, but this was from pooling samples across the entire year. So there's still a stone left to turn here, and that is temporal barriers to connectivity. So Winter Flounder produce more than one cohort per year in certain locations on Long Island, New York, and these are called split cohorts. So there's maybe, you can see in this little graph of spawning frequency in the seasons, you can see there's 2 peaks, and those peaks are different cohorts. So you might see a peak of juvenile abundance in the spring and a second peak closer to the fall. The question here becomes, are these cohorts produced by separate spawning modes? So back to our hypothesis of there being separate migrant and resident genetic lineages, which we sort of disbanded from the previous results we just talked about. Are these are these different split cohorts representing different spawning modes? So maybe migrants are spawning at different times than residents, or maybe there's 2 groups that don't correspond to migrant or resident, but they're spawning at different times. So they might are these different early and late cohorts genetically related to each other? So why is this important? It's because producing multiple cohorts within the same year is another example of life history diversity that can act as a portfolio effect. So if the conditions are unsuitable early in the year, then maybe the later cohort has a better chance. And if the conditions are unsuitable later, maybe the earlier cohort could grow fast enough to overcome that. And if these cohorts are part of the same spawning population, so that's something that we're trying to look into here. But we do know from previous work in Long Island, New York, and from work by others such as Susan Sogard in other locations, that bays that produce multiple cohorts have a higher post-settlement survival overall. And that survival of early versus late cohorts differs between years. So here's just a admittedly cherry-picked example of 2016, where we have, in 2016, we had the early cohort in this light grayish color having better survival than the late cohort. And then when you look into 2017 data, there's a total of 4 lines because there was a death interaction, but we have regardless of depth, we have the early cohort doing worse than the late cohort, which is the opposite of 2016. So we wanted to know if resident and migrant individuals might be producing separate cohorts. And again, we were able to piggyback on another study. This study, led by Catherine Ziegler, was an acoustic telemetry study of adult winter flounder in Mattituck Creek, New York. 72 adults were tagged and were categorized as she categorized them as either migrant or resident based on their behavior. And then simultaneously, beam trials were conducted in Mattituck Creek, which revealed two cohorts. Early young of the year spawned between December and February, and late young of the year spawned from March to April. So we have migrants and residents in Mattituck Creek, and we also have early and late cohorts happening in Mattituck Creek. When I look at genetic data from the study, what we see when we look at migrant and resident individuals is that they are indistinguishable. So we have the migrant adults and the resident adults, and they look the same. And then we have the young of the year, the early and late cohort, and they are the same as one another. They're very genetically similar, which is interesting. Maybe some sweepstakes recruitment happening, but they are similar, and the adults are similar. So another way to look at this is to infer putative family relationships in a program called Colony. So we're using differences in allele frequencies and those patterns to sort of create a pedigree for each potential family relationship. So the x-axis here is representing putative parents, with each tick representing an individual. And the y-axis represents the number of offspring they produced in each period that we encountered in our beam trial. They could have produced many, many, many more, but we're talking about the ones that we sampled. And then we have different boxes, different panels from top to bottom that are representing different cohorts. So we have 2015 early, 2015 late, 2016 early, 2016 late. And what you're seeing here is the same individual is contributing both early and late cohorts in 2016, and it's contributing to multiple year classes. So it's contributing to 2016 and 2015. So this is sort of disbanding the idea that these cohorts are early and late cohorts, the result of different spawning groups. And we were able to replicate these findings in Shinnecock Bay, even though we didn't have actual data on adults to compare them to. In this case, the parents are just created as part of the pedigree. They're inferred parents, but still we found that these inferred parents are also spawning across multiple years, spawning across multiple cohorts. I just highlighted one example, but if you looked at this closely, you'd see there's a couple more. Okay, so the bigger picture here is that we have characterized a lot of behavioral diversity that is leading to genetic structure, genetic diversity, and connectivity in the genetic architecture overall of these populations of winter flounder in Long Island, New York. So we have here we have adult-mediated connectivity in the form of straying between natal bays, which is serving to maintain genetic connectivity and reducing inbreeding. And then we have spawning habitat and migration propensity that is not driven by genetic differences in either neutral loci or loci under selection, or selective pressure, and that we haven't detected any pattern so far. So we could look, deeper into the genome and see if we could find patterns in other locations that we didn't sample, but we haven't detected any pattern of genetic structure driven by migratory type or nursery habitat so far. And then We also found that early and late season cohorts are not comprised of genetically distinct lineages, which is really important for the whole portfolio effect of cohorts. And the fact that like these bays that have more than one cohort are persisting longer into the season, it's really good news to find that these cohorts can't be individually wiped out because they are part of the same group. So ultimately, what we have is diverse behavioral patterns in spawning and migration, preserving genetic diversity and connectivity, and which is adding to the protective portfolio effect for winter flounder in southern New England. So I'll use the remaining time I have to offer some food for thought. One thing is population structure can exist in the absence of genetic structure. There's a whole literature on how that happens. And this means that genetic tools alone are not a sufficient determinant of stock structure. You can't just use genetic tools. You have to use a whole suite of different tools to understand this fully. Not only does defining stock units have far-reaching management implications now that we have greater power than ever before to parse groups genetically, and this is going to force us to really think about what the practical definition of a stock is. Like, just because we know there's this genetic structure, how relevant is that, and to what extent is that operational for us in managing these species? We have to think about that carefully. And then finally, sometimes we have conclusions from different methodologies that conflict, and that's something that highlights the importance of collaborative studies and of groups of different people of different expertise coming together so that we can sort of figure this out all as a team. Oops. So with that, I want to say thank you to you all for being here today. I know it's been a wild ride the past day and a half, so I really appreciate you coming to this talk. And I want to really say thank you to my co-authors, everyone who helped me in the field, my lab colleagues, and my funding sources. This was funded by and Saltonstall Kennedy grant, which has been a great tool for promoting research on, fisheries science in the region, and a New York State Wildlife Grant. And here you can see some of my collaborators and lab mates, including Shannon O'Leary, everybody in the Frisk lab, the lab of Michael Frisk at Stony Brook University, everyone in the lab of Anne McElroy, also at Stony Brook University. And then a lot of the genetic work was done at the Chicago Field Museum with the Pritzker DNA lab. So with that, I'll say thank you. And oops, we're going to the back slide. Okay, here we go. I'll say thank you, and I'll take some questions. Thanks.
Thanks, Tara. That was very good. If you have a question for Tara, please unmute yourself and ask the question, or you could put something in chat. Steve.
Thanks, Gary, and even more thanks to Tara. Tara, great job in the seminar. It's been great working with you on the flounder assessment working groups, particularly Winter Flounder stock ID. The work that you and your collaborators have done have really improved upon kind of a rich literature on winter flounder. So great job. I really love your 2024 genetics paper, but I agree, it's not all about genetics. We need to integrate that with other things. My biggest question is from a paper that you were not a co-author on, but your classmates were, is Matt Siskey's otolith chemistry. And thanks for explaining something that's been a question to me is, how can we classify 35% of the samples sampled on Georges Bank as Bay residents? And as you described it, they interpret that as straying, that some of the same chemical signals in the otolith may be from fish straying. But my question remains, how can a Bay resident stray onto George's Bank? What am I missing?
Yeah, you and me both, Steve. And thank you for those compliments about our work, especially to my collaborators, which I couldn't have done this without. But as you said, it is like very confusing. I did look into this a little bit, and besides the explanations that were offered in the paper, which was, as you said, straying from southern New England, and the other explanation that was offered that is out of my depth to answer is the idea that there could be hypoxia on George's Bank, which could indicate which could enrich the magnesium, which would make it seem like they were born in a bay. But that wouldn't explain the residence status, which is determined by the strontium calcium ratio in the entire otolith. So I did look into the toe that a lot of the different toes that these fish were caught. They are on the very, very southeastern edge of George's Bank. Very borderline George's Bank. So it did occur that if they were straying, I guess they cluster with southern New England, so it would make sense that they would stray from southern New England. But I think that we cannot rule out the idea that there is bias sampling, or even perhaps more worryingly, sample mislabeling, which has plagued me in the sense that I can't rule it out. So that's why I haven't actually published this work yet, because I'm still trying to think about whether this study is even publishable given that discrepancy. But if you have thoughts or advice on that, I'd be happy to hear it.
No, that helps. The other part was I think that was a low sample size
Very low, yeah.
Sample as well. Great job.
Yeah.
Thanks, Steve. Adrian.
Thanks, Tara. Really appreciate all the work and lots of food for thought, more than I'm going to be able to get to today. So I'll just, it's interesting to think, one of the things I worry about a little bit is our assumptions about how mobile certain species are. And I think that, when you're a flatfish, the automatic assumption is there's not a lot of mobility, but we've seen a lot of work come out about how these organisms can use tidal currents and other things to move around in a way that really enhances their ability to have connectivity. So not sure if that helps. I mean, I have no answer for why George's Bank has bayfish, but I'm also sometimes I'm just not surprised also, and it just leads to that. Anyway, I didn't have a question about that. I have a tougher question, and just when as a manager, you try to set rules in place that allow there to be both the maintenance of the stock over time, and I think importantly, as you pointed out, the maintenance of these portfolios of different life histories. So, put the manager hat on, and I'll ask you an impossible question, which is, your thoughts on how we can approach managing species where there are these different life histories that expose them to different agents of mortality at different life stages, and expose them to different levels of fishing effort, different types of fishing effort, both commercial and recreational, and just your general thoughts about how we can sort of move management forward to sort of accommodate these life history variants.
Yeah, thank you for that question. That's a good question, Adrian. I don't know that I could solve this problem, but I think that one heartening thing is the finding of this portfolio effect, that even if some stocks are disproportionately subject to fishing pressure, these other population components could rescue them. But as you said, we can't erode population components entirely, because then there'll be nothing left to save them. So I think, unfortunately, or maybe fortunately, the answer is sort of an integrated management, because there's different life stages. And for example, for the individuals spawned in the bay, protecting the bay habitat, that's a totally different collection of different agencies and different actors and stakeholders to promote, healthy bays, good nursery habitat. That's a totally different group to some extent than the management arena that's controlling fishing pressure, and even differences between managing inshore stocks and offshore stocks, which is like a little bit more integrated than the difference between managing fish populations versus managing habitats. But I think you really need to think of holistic management across the lifestyle, the life history of the fish. You can't just protect one thing and not think about the other things because they're vulnerable to different forces at different stages.
Thank you.
Thank you.
Other questions? Chat. All right, I guess that's it. Well, thanks everyone for coming. We will have another seminar next week, and Brad Chase of our Diagemus Fish Program will be talking about something. I don't have this title yet. I'll send out a link on Friday to everyone. So thanks for coming.
Thank you, Gary. Thanks, everyone.
All right, talk to you later.
Bye.